Good morning, everyone. Thanks to the organizers for the chance to speak today and for putting together what has been a really exciting couple days of talks and of science. So I am going to be continuing the trend at this conference of really great Drosophila work by talking about mitotic waves in the developing Drosophila blastoderm. But before I get into Drosophila in particular, and before I start maybe showing some movies of this really exciting developmental process, I'm going to start by talking about a more general problem, which is about changing density and densification. And so I have here a couple of really striking examples in nature of where we see major changes in density that lead to changes in structural properties of the system. Changes in density are important in many natural cycles, and when we study changes in density, we think about not only what the density was before and what the density was after, but how the material transition from those states. So, at a overly abstract level, thinking about increasing the density of a system is very simple. If we treat density as the number of particles in a system divided by the volume of a system, it's not too difficult to think of two clever ways of increasing the density. One of them is to decrease the volume, and the other is to increase the number of particles, right? But of course, this is it's a little bit more complex than that. And what really matters is the algorithm that you implement to go from low density to a high density. And so one of these first examples that I'm going to show is back to the magma that I had on the very first slide. When you have particles that are condensing, in the case of magma, you can cool those particles down very slowly and give the particles enough time to rearrange into a nice crystal lattice where you'll end up with long-term structure or long-distance structure and organization. In contrast, if you cool the system down very fast and the relative time scale of rearrangement is a lot slower than the time scale of cooling, then you end up with a structure that still has some local organization, but a lot less long-term organization. You get something more glassy, like volcanic rock, or volcanic glass. So this was an example, right, of the relative time scales being important when you're decreasing the volume of a system. The other example I'll show here is that when you're increasing the number of particles in a system, the algorithm that you choose to use to inject these particles in the system is important. If you look on the left here, we have the maximum coverage that disks can fill up the plane. This is about a 90% coverage in this hexagonal grid. But one sort of intuitive algorithm that you can imagine for adding new particles into a system is to sort of randomly place these particles, and if the random placement that you choose intersects with an existing particle, then you remove it. If it finds its own space, then you can keep it. And so as you shoot these, this is called random sequential absorption. If you sort of randomly shoot these particles onto a substrate and let them bounce off if they collide into something, or let them stick if they have their own empty space, and you continue this until there's really no more room left for a new particle to be added, you end up with only about 55% coverage compared to a much higher maximum possibility. And so, of course, this is all abstract, thinking about materials, but it is sort of connected to developmental biology in the sense that developing organisms are also undergoing a process of densification. And so when you start off life as one very big cell with just a single nucleus. And this is an example of very low nuclear density, right? This is, n is 1, and volume is big. And so at the start of life in embryonic development, you go through, with a constrained volume, pre-implantation or pre-hatching, you go through many nuclear divisions that increase the number of nuclei in your system while keeping the volume constant. And this is a really important densification problem, and that is sort of the problem of developmental biology, is to ask how you go from one cell to many cells, and to what extent the trajectory that you're taking in this densification algorithm is going to impact the structure of the resulting embryo. And so different model organisms have different extents to which you can study this problem. We're looking here at Drosophila, which is a really exciting and very tractable system to be able to investigate these early nuclear divisions in development. So for those who have not seen this embryonic system before, in Drosophila, these early cleavage divisions happen really fast. Over the first 2.5 hours of development, the Drosophila egg, which is about 0.5 millimeter long, goes from one single nucleus to around 6,000 nuclei. And so this process is extremely rapid and very interesting to watch. In the first 9 divisions, these nuclear divisions are happening inside the yolk of the embryo, but after these 9 divisions, nuclei move out towards the surface and go through 4 more divisions. And the reason we can talk about these as divisions and being able to count them is because these divisions are very nearly synchronous. And so they partition the early developmental trajectory into a couple of different, or 14 different nuclear cycles that are separated by 13 nearly synchronous nuclear divisions. And so the spoiler here is that these nuclear divisions towards the later end of this developmental trajectory, these last 4 divisions, which are happening at the surface, become more and more wave-like over time, whereas in the early divisions, everything is very synchronous. In these later divisions, you have waves of mitosis that propagate along the axis of the embryo, and so I'll show that soon. But I want to start off by saying that this is a system that people have been looking at for a very long time. These mitotic divisions have been very interesting since as early as 1941. I have this paper here on the left of a PhD thesis of a student who looked at thousands of Drosophila eggs as they were being hatched, just watched and counted these eggs as they got laid on the plate, and then after watching them for a few hours would pierce them with a needle and look at the number of nuclei, and found that this organization was split up into discrete nuclear divisions, discrete segments of division. And this was, of course, a very difficult process at the time. And as technology has improved and our ability to look at these systems has improved, we continued to look at these developing Drosophila, and in 1983 there was the observation that these later mitotic divisions are happening in waves. And so I'm interested in the sort of densification problem. I'm interested in how the implementation of these mitotic waves, how the injection of these thousands of nuclei into the embryo affect the resulting properties in the embryo. But it is worth mentioning that a whole lot of interest and dedication has been put into studying how the underlying chemical system makes these traveling waves possible. And so I'll shout out here the D'Italia lab down at Duke, who has really been a modeling powerhouse for these systems and has shown how the cell cycle feedback loops can lead to all sorts of communication and how these waves can be happening so fast across the embryo. And this is really worth a read. However, these existing studies have, or previous studies have really looked at the local properties of these waves, but mitotic waves are an embryo scale phenomenon, and it's worth looking at the entire embryo when you're studying these waves. And so that's what I'm showing here is that in our research we look at the entire embryo, all sides of the embryo as it goes through these mitotic divisions, and we tag histones to be able to look at these nuclei as they divide many times in a row. So you can see here, right, these are these waves I'm talking about, especially this in these very last divisions. You can see the wave really move from the poles in towards the center. These are nuclear cycles 10 through the start of 14 that we're able to image here. And so what we're going to get at with this full imaging system is to ask how the trajectory of these mitotic waves, how this nucleus injection algorithm actually traverses the embryo. So if this is an embryonic scale phenomenon, what paths does it take? We're going to ask how the mitotic waves might influence this final structure. And finally, we'll look at some genetic perturbations that manipulate these waves and ask how it affects the packing that we see normally in wild type. Great. So as I mentioned, we're able to image the entire embryo, and that's because of some really great light sheet microscopy that was done by Liu in the Schwarzman lab. And this light sheet microscopy, we are imaging all four sides of the embryo every 20 seconds, and we're able to stitch together these four orthogonal views to generate isotropic data of the entire surface of the embryo. And so this is some really high resolution data at a very fast time resolution, which is really necessary if you want to be able to look at every single nucleus and follow it through time. Right, so we want to, this is the final result of this densification process. It's these beautifully packed nuclei all across the surface of the embryo, and we want to know how these nuclei got here. And so since we saw it happen, we want to be able to take individual nuclei that are at the surface here and follow them backwards in time and ask where they started, what processes they went through to get where they ended up. Since we're, as you can see, there are a whole lot of nuclei here. This is a totally impossible system to be segmenting and tracking by hand, and so we implement automated segmentation and tracking for the system, which is really non-trivial. And so I'll talk briefly about what we do to get this result. So we're interested in things at the surface, and so we are looking at these nuclei, and one question you can ask when you're working with this system is what sort of data transforms you can do to make this problem of segmentation tracking more tractable. And so we have, on the left, you can see that if you look at individual slices in this 3D volume, there's really not that much information to be able to segment. On the contrary, if you look at a max projection of this entire data, you can have a lot more information, but there are issues with warping and overlapping and limitations on the segmentation. And so the end result that we've settled in is a is an not settled, but the end result that the process that we've developed is a full unwrapping of the embryo and a segmentation of this 2D unwrapping. And so there is a really great preprint out from the Striking Lab that allows you to go into Blender with meshes that represent your data and unwrap these meshes into UV maps and take those UV maps and or take the 3D volume and unwrap it into a 2D mapping. And so we import our meshes into Blender and we unwrap these embryos, and hopefully you can see on the left here that once you unwrap the embryo, it becomes a lot easier to see where these nuclei are. This is great data for segmentation with, we're using CellPose here, which is a 2D model. And once we've segmented in 2D, since we know how we unwrapped these embryos, we can project that data back into 3D, which is what you see on the right. So the last part of this segmentation and tracking process is tracking. We have a two-step algorithm that we've implemented here. The first step is a sort of linear tracking following nuclei through from time point to time point. And the second part is a sort of daughter to mother reconstruction. So we follow nuclei individually, and then at divisions, we have a separate algorithm for assigning daughters to their parents. You can see on the left here, I'll look at it for a little bit longer. These are nuclei colored by the founder nucleus that they came from. And so at the start, we have every single color is represented once, and then over these 4 divisions that we're showing at the surface, you end up with around 16 of each color. These are the great-granddaughter cells of our original nuclei. And so we're doing some work in the lab to understand a bit more about these individual lineage packings, but today I'm going to talk about stuff that we can learn just from following going from the starting positions of these nuclei to their end positions. We are tracking thousands of nuclei per embryo, and fortunately, every time a nuclear division goes by, we double the amount of nuclei along the surface, which is great. You can see, yes, at the end we have 5,000 to 6,000 nuclei that are fully tracked. And once we are able to track nuclei, follow them through divisions, we can see exactly when they divided, and we can split up our embryonic development into its different nuclear cycles. So I have here the nuclear divisions colored by what nuclear cycle, or the nuclei colored by what cycle they're in. It's a little bit jittery, but I'll show on the next slide. I have a couple of freeze frames of this very last wave. And what I want you to remember here is that there's not exactly a perfect wavefront, but you can see this trend of nuclei that start dividing. So yellow here is before divisions, and blue is post-division. What you can see is that these waves of division start at the poles and move inwards. And if we then take these nuclear division times, so I have in these plots here, each nucleus is represented by a scatter point on these plots. This is just looking at that embryo that we've already been watching in a lot of different forms. We can see that our points along the AP axis correspond very tightly to a particular division time. So this kind of confirms what we've been seeing, which is that the AP axis position determines when you're going to be dividing. In contrast, if you look at, rather than taking the AP axis, which is this, the AP axis is this long axis over the embryo, rather than looking at position along this AP axis, if you look at position around a dorsoventral unwrapping, what you find is there's not really a relationship between dorsoventral position and the nuclear division time. And so to really drive this home here, I'm showing that if we look at the nuclear division times and like this mitotic wave from all four sides of the embryo, what we see is that all four sides of the embryo are really undergoing the same AP position dependent mitotic division timing. Yeah. And I'll just show here, this is obviously a lot of plots, but each of these plots, yes, 5 minutes, yes. Each of these plots is representing a single embryo. What you can see is that as a function of AP position, even if the waves vary in what they precisely look like, this AP position specific relationship holds. And so, yes, the important parts of this, which are, I should mention, not particularly new, but we can really show this with a full embryonic view. We can see that mitotic waves traverse the embryo primarily along the AP axis. They move really just in this AP axis dependent way, and that these mitotic waves in the embryos that we've seen begin at the poles and move towards the center of the embryo. And so the questions we'd like to ask based on this are, what are these mitotic waves doing? Is there any functional role here beyond the sort of, everything is roughly finishing at the same time? What we see is that if we follow these nuclei through time, through these nuclear mitotic waves, and right here we have nuclei that are colored by whether they've moved more anterior or more posterior. What we can see is that these nuclei that are more anterior, that are starting off anterior, move more anterior, and things that are posterior move more posterior. And so what in effect this is a stretching along the AP axis where things move away from the center over the course of these four mitotic divisions. And I have here plotted on the left the sort of initial versus final configuration of these nuclei. Nuclei move around, can move up to 5% along the AP axis as a function of the stretching. And what you can see is it's this really embryonic scale trend where nuclei are being stretched along this AP axis. On the right, I'm showing maybe what the functional role of this, which is that in nuclear cycle 10, at the very start of this video, you see a not very uniform density profile of nuclei along the surface of the embryo. And by the end of this, so in particular, the poles, which has been known, the poles have a much lower density in the center of the embryo. And over the course of these nuclear divisions, we sort of produce a more uniform density profile along the anterior-posterior axis. And so this is sort of a correlative relationship, and the last thing I want to show is one way that we can perturb the mitotic waves to be able to see how that affects the resulting structure. So just briefly, there are developmental cues in the embryo that are pre-patterned by the mother. One such developmental cue my lab has shown is able to influence mitotic entry. And so the idea of this is that ERK, which is present or is active at high concentration at the poles, may be helping to speed up mitotic entry at the poles. So if we remove this mitotic gradient, if we remove this developmental gradient, the result that we see is that so now we've removed this high concentration of ERK at the poles. What we see is that the mitotic waves are much slower to reach the posterior. And so hopefully you can see in this video that on the posterior on the right, that these mitotic waves are really slowly getting to the posterior. We have that quantified here. These are very, very slow waves. Rather than starting at the poles, they start off the poles and move towards the pole. And the very last thing that I want to show is that possibly as a consequence of these unusual mitotic waves, what you see is that the density profile of these embryos without this developmental gradient is a lot less uniform than those in the wild type. And so this sort of model here that is ongoing work and that we're working on in the lab right now is to suggest that the direction of these mitotic waves is in some way influencing the density profile of the embryo, and that when you have mitotic waves that start off the poles, you don't end up with nearly the same uniformity in the density profile. And so just to conclude here, so we've shown a densification algorithm that forms a blastoderm, and the way that we were able to study this formation was by looking at individual nuclei. So this was a densification process, and we benefited the most from being able to really look at this process in detail, follow the trajectory through time. And what we found was that these mitotic waves are not only injecting thousands of new nuclei into the surface of the embryo, but they are simultaneously organizing density at the embryonic scale. And so with that, I'd like to thank some people at my lab and people that we've been collaborating with. Leo, of course, who's been doing the imaging, Hayden, who's been really great help throughout this process and has helped me with getting resources to be able to run these GPUs and do all this segmentation. Stanley, who's was working at the Flatiron Institute and has been perusing this data, and we're continuing to collaborate for a while, and Daniel, who's been great help with the image fusion and working with these large data sets. Thank you to the Flatiron Institute and to NITMB, and thank you to all of you for this.
Questions?
Very nice presentation. I have a question regarding the onset of your waves. Could there be a curvature effect in your lattice? The reason why I'm asking is I'm working on viruses, and HIV particles have these cone structures, and we have this unraveling of the cone for release starting also in waves. These gaps start in waves at the highest curvature points. So is there any relation?
Yeah, it's a very good question. So we've made, we made changes to the embryo by removing the developmental gradient, or this gradient of ERK at the poles, which was able to sort of shift the waves. But naturally, there's a question of geometry and curvature. I mean, these are these are not just there are boundary conditions in this system. So we haven't really looked at the curvature between these different embryos doesn't vary too much, and so it's there's a limit to how much we can ask about the curvature in this case, other than some sort of correlative stuff. There's more curvature at the poles, and we know that these mitotic waves are starting at the poles, so that could absolutely be a relationship. We can look at other Drosophila species or different insect species where you'd see different shapes or different size of the egg, and maybe be able to drive that relationship home a little bit better.
Thanks very much.
Thank you.
Very cool talk. So I had a question about your 2D representations and the tracking. So it looks like the poles at the AP axis are really important, but the 2D representation, I'm curious how when the nuclei are actually moving to the pole, if your tracking kind of fails at that point, or what's the capture at the pole of nuclei once they move?
Yeah, that's a good question. Yeah, so for a project that's very related to what's going on at the poles, and that is, sort of the major benefit of being able to look at these embryos across the with this light sheet microscopy, it's compared to confocal, where you're only able to look at sort of some of the less curvy parts of the embryo. Being able to look at the poles is really important, and this light sheet microscopy makes it possible. So yeah, we had to address that when we were doing the segmentation and tracking. The unwrapping that you saw actually is a little bit of an older version of our unwrapping. And so in that older version of the unwrapping, right, it's just split into 3 different parts, where the poles are this little nub at the end. And yeah, that leads to issues with tracking, because during the segmentation, like if things get cut off by the seam, like you can have all sorts of issues. And so the way that we've resolved this now is we split the embryo into like a sort of dorsal patch, a ventral patch, and then a patch that covers a wide region of the poles. And these patches are overlapping. And so we segment these nuclei in such a way, or we split this embryo mesh up in such a way that there are large overlapping regions, and the segmentation is able to perform well outside of like these seams. And so we can, with very high accuracy, we can take 2 overlapping segmentations and sort of co-correspond to them. And so we no longer have these seam issues that you would imagine if you just take the embryo and unwrap it once.
It's very interesting. I was wondering, for your last part, what's the mechanism for slowing down of the wave? If I understand, the terminal speed of the wave should only depend on the property of the medium that wave propagates. So I wonder if the slowing down you see is because the phase wave to the terminal, the trigger wave speed, that when you quantify is if you capture the terminal speed.
So the question you're asking is how you're able to see such a drastic change in wave speed with this genetic manipulation, is that right?
Yes, so if so for your Drosophila system, initial early cycles, you have very synchronized wave, right?
Yep.
But those waves are characterized as a phase wave because there's no time for them to really connecting, right, for the interaction yet. So I guess my question maybe more directly is, how do you quantify the wave speed? Do you always quantify until it gets to the trigger wave regime, or?
Yeah, that's a good question. Yeah, so that is sort of what the Tatalier lab has been looking at for a long time with the Drosophila system is these wave speeds. How are the wave speeds that you see possible? And they developed a model that initially proposed a trigger wave that was propagating along the AP axis, but a trigger wave would just be too slow for the kind of wave speeds that you see in the healthy embryo. Even in nuclear cycle 13 to 14, when these waves are at their slowest, they're still faster than you would expect in a trigger wave regime. And you can kind of see, so really the model that's that they've kind of settled with is, these are sort of phase waves where there are sort of position-specific delays. But what you see in the truncanal embryos that I was showing there is, you do see a very, very slow wave that reaches the posterior, and I quantified this wave speed just, because it was somewhat linear and because this wave speed was so clearly in the more trigger wave regime. And so it seems that whatever is going on in that posterior pole is in some way affecting that sort of autonomous or non-autonomous underlying chemical system. And so, we switch over to the trigger wave regime for one reason that we haven't totally determined yet. Thank you.